Hello, this is a short talk to illustrate the statistics in genomics and microbiome that are happening in the lab. And these types of models are all built around high dimensional data, where we have more columns in our data matrix X than we have rows or sample. And our goal remains the same. We want to estimate beta that will identify which of those columns have an effect on the response Y. And there are many examples where this type of data will arise. For example, we have microbial genome studies where we want to study the emergence of antibiotic resistance. In this case, the Y corresponds to some measure of antibiotic resistance, and the X are different genes in the bacteria. Similarly, we can do human genome studies where the X corresponds to genes, but now genes on humans, and the Y can correspond to any response on humans. Similarly, the X could be the presence or the abundance of different microbes, either in the gut or the lung or the skin or different parts of the human body. And lastly, another example is the soil microbiome studies where our X corresponds to microbes that are present in soil samples, and the Y is some measure of plant disease susceptibility. These are examples, by the way, of all current collaborations in the lab. And the challenge, one of the challenges is that aside from the fact that it's a high dimensional data, is that our samples tend not to be independent. So in the case of microbial GWAS, the bacteria in our sample, they are correlated because they have all been evolved or cloned in the lab. In the case of human GWAS or human studies, it could be that you have familiar correlations in your data. Or lastly, if you have soil microbiome data, there could be some spatial correlation depending on how these samples were collected. So our goal is to incorporate this correlation structure into the sparse learning theory. And one way in which we can do this is via post-selection inference. And in this case, there is it's a combination of doing inference on beta, that is estimating beta, but at the same time doing model selection, because we want to also identify which are the betas that are different than 0 at the same time. And there is a really interesting paper already about post-selection inference when you have lasso as the penalty. And that basically answers the questions of what is the asymptotic distribution of the estimator of beta under a lasso penalty. What we want to do, and currently this work assumes that all the samples are independent. What we want to do in the lab is to extend to different dependent structures and also to other types of penalties. And this is a type of work for students that are interested to do research in the lab, that it has either very theoretical foundations of mathematical statistical theory or implementations via software or computational tools, and also applications to real data, as I described in the previous slide. And this is not just the sky dimensional setup. I mean, we can analyze through this regression or post-selection inference, but when we are dealing with microbes specifically, there are other things that we want to do. In particular, we might want to understand the interaction between the microbes in addition to how they affect a response. And this is another type of research that we do in the lab. We want to estimate a microbial network that represents the interactions between different microbes. Here, each node corresponds to a microbe, but also not view it as a static object, because we know that this microbial network, it can be changed by either environmental forces or experimental forces in the lab. So there's really 3 levels of knowledge that we're trying to build. So we have the microbial network in the middle, but we also want to understand how the environmental or experimental forces affect the network. And in turn, how does the network structure and all this interaction affect the plant phenotype or the human phenotype? And I'm just gonna briefly mention 2 projects that are happening in the lab, or I mean, they're ongoing. So one by master student Jinji Shen, he created an R package, and also there is a preprint already here for you to check out, where we implemented a Bayesian Carl-Lasso model where we reconstruct a network that has 2 types of nodes. Triangular nodes correspond to predictors, let's just say antibiotic or humidity or something that is affecting the microbes. And the circle nodes, they correspond to the responses. In this case, it could be microbial abundances. The edges encode conditional dependence, and the color represents the direction. So either it's positive or negative. And basically, we have two types of matrices in this model. The B matrix is the adjacency matrix for the predictors and the responses, and the omega matrix is the adjacency matrix among responses. And this is just a reparameterization of a standard normal model. But by doing this parameterization, we're focusing on edges that are conditional. And in this way, they align better with the intuition from experimenters. There's a huge discussion about that in the preprint, so make sure to check that out. And another example is a Bayesian network regression being implemented by master student Sam Osminkowski. And here, the difference, we are building on a model that has been published by Guha and Rodriguez in 2020, where the predictor is the adjacency matrix, and we want to know how it correlates to a specific plant phenotype. This model is able to estimate or to infer which are the influential edges and which are the influential nodes that have an effect on the plant. Unlike the Guha and Rodriguez work that they do this paper for brain networks that are not sparse, we are extending this model to the sparse networks that arise from microbiome research. And Sam is working not just on the model, but also on a Julia package that's been implemented. So just as usual, the microbiome research that is happening in the lab, it has a strong mathematical or statistical component, but as well, it could be, it will have a software or a computational implementation, and everything is really connected to applications or biological applications